Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Habib. I'm here with my co-host, JP, and we are here with a very special guest today, Dr. Thomas Seyfried. It's wonderful to have you here, Dr. Seyfried, and we are honored by your presence.
Oh, thank you very much. It's a pleasure to be here.
So we're really interested in talking about the autonomic nervous system, longevity, and in your situation, we want to talk about the metabolic basis of cancer because as your research has shown, there are very unique factors when it comes to understanding why cancer exists in the first place and what can be done about it outside of what we are traditionally looking at cancer as. I think there's been a very significant push that cancer is a genetic condition, and your work has proven that there is a metabolic basis. And I want to get into what that is today. I want to first start with your kind of journey because you started in looking at epilepsy as your major field or major condition of study. How did you move from the epilepsy world into understanding cancer, and what was that journey like?
Well, I actually started doing lipid biochemistry and genetics, but when I was at Yale University Department of Neurology, it was clear that if I wanted to stay, I better find something to do with epilepsy, because the whole department at that time under Gilbert Glasser was a leading department in the world for epilepsy research. So I decided to try to map genes for audiogenic seizures, and we published some major papers trying to do that. And while I was at Yale, I did think about ketogenic diets for managing seizures, but I wrote an internal grant to the department medical school, and they said nobody's interested in ketogenic diets, passe. They were using drugs, and I didn't think much about it. But then, we developed some of the best animal models of epilepsy. And when I left, I was on the faculty at Yale as an assistant professor. But then I left Yale and I came to Boston College and started my genetic analysis of epilepsy. And we were mapping genes and characterizing the phenotypes of these different seizure models, epilepsy models. And one of my students, Mariana Tran, got invited to a meeting in Seattle, Washington, by Jim Abrams, the movie producer. He made the movie Airplane and a few of these others. His son Charlie was rescued from a near-death epileptic seizure death by the ketogenic metabolic therapy at Johns Hopkins. And Meryl Streep did the movie First Do No Harm, which was based predominantly on Charlie's experience, Jim's son. And Mariana came back and said, "Oh man, everybody's excited about ketogenic diets and epilepsy." And I told her, I said, "Nobody cares about that." And she said, "Oh, no, there's a lot of new stuff." So we started to take our mice and put them on ketogenic diets. And we were able to show that a lot of the success of ketogenic diet was actually calorie restriction. And it was a lowering of blood sugar and elevating of ketones. But at the same time, we had a big parallel study of cancer, brain cancer, but purely for biochemical reasons, not for any kind of therapeutic attack on this. I mean, we were just looking at differences in glycolipid biochemistry between normal brain and brain tumors. And then we started to get a little bit into the angiogenesis studies. And angiogenesis is abnormal vascularization of the tumor. And we were, one thing led to another, and we were putting some of the tumor mice on calorie restriction, and we were able to show tremendous reduction in angiogenesis and shrinkage of the tumor and a variety of other observations. And then I said, what's going on here? And it looks like the way calorie restriction and ketogenic diets work for epilepsy was a lowering of blood sugar and an elevation of ketone. And when we started on the tumor, we saw the same thing: lower blood sugar, elevated ketone. And then that linked me into what Otto Warburg had said, which was the famous German scientist from the 1920s up to the 1960s. He was saying cancer cells live on sugar because they have mitochondrial dysfunction. So we started looking at that, and if they have mitochondrial dysfunction or they're dependent on a fermentation metabolism, then it would explain why we were getting the really spectacular results that we were seeing. So we started to do deep dives on signaling inflammatory markers. We started to look at these cancer signaling cascades, and we were seeing them all downregulated under calorie restriction, lowering glucose, elevating ketones. So then we said, well, what about, and then we were doing it with water-only fast. So the problem with water-only fasting, a 40% calorie restriction in the mouse is comparable to a human doing water-only fasting. Well, we were all excited about how powerful this was, but we felt that we would all be considered nuts and crackpots if we were telling cancer patients to go out and starve or stop eating, because the physicians think that, oh, you got to put as much weight on the cancer patient, mainly because we're giving them toxic poisons and irradiating them. They're going to lose weight, so they better eat as much carbohydrate as they possibly could. And I was saying, well, geez, that's just the opposite of what we should be doing. We should not be giving them carbohydrates. So, but then, to this day, many oncologists think you have to pump the cancer patient full of carbohydrates to keep their weight on. And I'm saying, why are they losing weight? Well, you're being poisoned by toxic chemicals. You're going to lose weight, right? You shouldn't be doing that to the person in the first place. So you should really be looking at the nature of the problem. So one thing leads to another. And then we started to look deeply at the mitochondria number, structure, and function. And we found them to be all abnormal in cancer cells, regardless of the type of tumor. That means we knew that they could not use oxidative phosphorylation effectively. And then we supported Warburg, showing that they were fermenting. But our new big thing that Warburg did not know about is that he was all, can't use oxphos, cancer cells upregulate glucose-driven fermentation, and that led to a big brouhaha because it looked like some tumor cells were taking in oxygen. They didn't. They looked like they had normal mitochondria, and this problem exists today. We have clearly shown that they can also ferment an amino acid, glutamine, in the mitochondria, which is brand new. I had speculated on that in my book in 2012. I speculated on it, and we put a map together on how it might occur. But now we've really fleshed it out with my work with Christos Xenopoulos and Semmelweis University, world expert on mitochondrial substrate level phosphorylation. And we showed that it was glutamine was the primary fuel. And everybody I shouldn't say it, but almost everybody in the cancer field knows glutamine is a prime fuel for cancer. The problem is they think it's respired. It's not respired, it's fermented. That's the new thing. It's an amino acid fermentation. So we started putting everything together. We saw where Otto Warburg was incorrect. His concepts were absolutely correct, but the way he tried to demonstrate with quantification of oxygen consumption and lactic acid production linked to how much ATP could happen from both of these processes was incorrect. And bottom line, that led to the big arguments about the Warburg hypothesis, and he was wrong, and blah, blah, blah. And everybody was running off chasing genes, and very few people, he was more or less abandoned. And of course, toward the end of his life, he became unhappy about the fact that he was the big guru for the 20s and 30s and 40s. And then all of a sudden, he's thrown under the bus. Nobody cares about him. And they ran off chasing genes, which they're still chasing in the field. The National Cancer Institute says cancer is a genetic disease. Nothing could be further from the truth than that statement. It is definitely not a genetic disease. As a matter of fact, most of the genes for cancer are irrelevant. They have no relevance.
Can I just jump in here? Because one of the things that I'm a big fan of your work, and I actually totally agree with what you're saying. But I haven't heard anybody make this argument, and I'm wondering what you think of it, which is if you look at all of the different mutations that the National Cancer Institute and researchers who are focused on the somatic mutation theory of cancer, if you look at what those mutations are, they are largely mutations of proteins or genes that code for proteins that are critical for mitochondrial function. Remember that most of the mitochondrial genome when mitochondria were originally, subsumed into bacteria and became the symbiotes they are today, most of their genome actually transferred over time into the nucleus.
Yeah.
And the nuclear transcription of those proteins is critical for mitochondrial function. So if there's a dysfunction in the mitochondria, it is possible that the reason why you're getting that mitochondrial dysfunction is because of a genetic mutation in the nucleus. But look at things like BRCA1 and all of these semi-penetrant, they're not 100% penetrant, I recognize, but these semi-penetrant genes that seem to increase your risk of cancer, if you dig down into what those genes are supposed to code for, they're coding for proteins that have a key role in making certain that mitochondria can function properly. So if the mitochondria have the ability to function properly because they're being taken care of otherwise, they might be able to withstand this mutation. But in the end, I agree with you that it is not due to that mutation. It's due to the effect that mutation has on the mitochondria.
I think that can explain some of what we see. But we also looked at the nuclear mitochondria transfer experiments. So when you take the nucleus out of a tumor cell that has all of these mutations that are coding for whatever, and put it into a new cytoplasm, you get regulated growth. You don't get dysregulated growth. And if you do the opposite experiment like Israel and Seyfried did, and this has been done in frogs and in mice and these kinds of things, you take the nucleus of a normal cell and drop it into a tumor cytoplasm, you get dysregulated growth. So clearly, the mitochondria are calling the shots on this whole disorder, and the genes are largely downstream effects. And when I say they're largely irrelevant, the nuclear mitochondrial transfer experiments pretty much slam the door on that. Now, you're right. There might be a gene, and we looked at every one of the germline genes, not just BRCA. We have P53. We looked at the Lynch syndrome. We went through and looked at all 15 what we call cancer syndromes, and every one of them affects mitochondrial function in some way. So, and none of them are 100% penetrants. Li-Fraumeni is the highest. It's about 90%, but there's still 10% of people that have the mutation that never develop cancer. So in order for something to be a prime cause, there can be no intervening disruption between mutation and phenotype. And that's why none of them are 100% penetrant, meaning none of them are primary causes, they are secondary causes. Anyone that would have a tumor as the result of a germline predisposition has mitochondrial dysfunction. We have never found a tumor anywhere that has normal number, structure, and function of mitochondria and is not fermenting. So that tells us right there, no ambiguity at all. This cannot be a genetic disease. It's a mitochondrial metabolic disease. But
it's just that the genetic mutation that is the secondary cause, not the primary cause, is actually working through, in many cases, disabling or otherwise contributing to the dysfunction of the mitochondria.
Yeah, making the mitochondria the primary origin of the disease.
Exactly.
All right, so let's clear that up.
Yep.
Right. So I don't want to hear any more. I mean, not from you guys, but people are, you got this gene, you got that gene, you got the RAS gene, you got this gene. Now we're finding that all these so-called, we separated the genes. We used to call, cancer is a genetic disease, but then we found that a lot of the genes don't have anything to do with cancer. But then we wrote in a terminology of driver genes. Okay, the driver genes are the ones that are really the important ones. Well, today, more and more papers are coming out showing our normal cells are full of driver gene mutations. P50, there's no cancer associated with this, right? And one other thing that the genetic guys don't like to talk about are the cancers that have no mutations. You got to love that. And when you show them the data from papers that have been published, they don't believe it. And I'm going to say, what don't you believe here? They looked and they didn't find any, or they didn't look hard enough. And I said, well, they used the same technology to show mutations in hundreds of other similar cancers, but not in a few of these. How do you explain that using the same technology? Some cancers have no mutations.
Well, let me ask you this, because I've been doing some work in longevity and looking at the hallmarks of aging. And of course, cancer rates go up just the same way, just as mitochondrial dysfunction seems to go up with age. But the connection that I'm making, and help me, understand from your perspective the viability of it, is that it's not genetics, but there is an epigenetic component to it, because you see in a lot of cancers, you see proteins that shouldn't be in that tissue type being expressed. And that suggests to me not a mutation. It suggests to me that the epigenetic controls on whether or not that protein is being expressed have been lost. And the way you lose epigenetic controls, one of the primary drivers of loss of epigenetic control, are free radicals. And the free radicals are coming from the mitochondria.
Yeah.
So I actually think that mitochondria aren't the downstream consequence of, the dysfunction of the mitochondria isn't the downstream consequence of a mutation. It's the upstream driver of the epigenetic control loss that leads to aberrant protein expression. So when you see that aberrant protein expression in tumor cells, it's not about the mutation, it's about the epigenetic control loss.
Yeah,
and that's from mitochondria.
You're absolutely right. And what I said in my big paper, don't forget the mitochondria is the most powerful epigenetic system in the cell.
Absolutely.
So we're learning more and more that our destiny, longevity, is all linked to the structure and function of the mitochondria. It's calling the shots in our cells, and we die when we run out of energy, and we have systemic failure of energy, and that's coming from the mitochondria.
That's right.
And second law of thermodynamics, entropy, disorder. When the mitochondria can no longer produce enough energy to maintain the membrane pumps, sodium, potassium, ATP, and magnesium ATPases, which maintain an electrical charge across the membrane, we go into equilibrium and we're dead. So why are we not dead? Because the mitochondria maintains the pumps. The pumps keep us alive. When the pumps fail, we're gone. And who's maintaining? It's all mitochondrial function. And not only that, they maintain the differentiated state of the cell. And what happens in cancer, don't forget, there's a two-step process. It's a chronic interference with mitochondrial function, efficiency of oxidative phosphorylation, coupled to a protracted increase in fermentation metabolism. And the maintenance of the fermentation metabolism is the upregulation of oncogenes, which are facilitators of bringing in the fermentable fuels, glucose and glutamine. They need to open the floodgates because efficient OXPHOS is so highly efficient. You can get so much energy with efficient oxidative phosphorylation. When that efficiency becomes compromised, we fall back on a highly inefficient system. And that inefficient system has to have a lot of logistics, a lot of supply of the fuel to make up for the inefficiency of the respiration, making up for how efficient it was. So oncogenes to open up the floodgates for glucose and glutamine transporters coming in now, because the mitochondria can no longer produce enough energy to maintain the pumps. And when they do that, the mitochondria loses control over the destiny of the cell. The cell falls back on its default state, which is proliferation, driven by a fermentation metabolism. So this beast is simply an energetic non-oxidation using fermentation metabolism. And fatty acids, ketone bodies cannot replace, they're non-fermentable fuels. So the strategy for managing cancer is not that complicated. You have to take away the two fermentable fuels while transitioning the whole body to nutritional ketosis, keeping the normal cells in the brain and heart and muscles completely functional and marginalizing the tumor cells. And then you come in with glutamine targeting drugs pulsing, and you just absolutely slaughter these tumor cells in a non-toxic way, and you allow your immune systems to come in and pick up the corpses, because you can't be too aggressive in targeting glutamine, because the very immune cells need the same fuel the tumor cells need. So that's why you have to know the biology of the beast and the normal and abnormal cells that are participatory in the progression of this problem.
And you want to make certain that those immune cells, when they're coming in to clear out the debris, they're doing it in a non-inflammatory way.
Yeah.
Because if they come in an inflammatory way, then they're going to be shifting over to that same fermentable fuel-based as opposed to relying on OXPHOS and fatty acids, et cetera. So listen, I think it's worth going back for our audience and just because you just gave us basically the heart and soul of the press pulse method.
Yeah, and not only that, I gave you the future of how you're going to manage cancer.
Well, and I agree, but I think it's worth going through and maybe discreetly providing this in a slightly more digestible way for our audience, just because you know this so well. I'm pretty well versed in it, having read and listened to all the teachings that you've had on this. But I think it'd be really great for the people who are new to this to understand what you're saying, which is that cancer is likely not, it really isn't. All the evidence seems to show that it is not the result of a mutation in the genome. It is a result of a dysfunction of the mitochondria. And that when the cancer cells become cancerous, their mitochondria lose their ability to function, and they have to rely for energy on a more primitive way of generating energy, which is less efficient. And so it requires a lot of dedication to that cell to bring in the fermentable fuels, glucose, and your discovery that glutamine can be used as a fermentable fuel and glutaminolysis to generate energy for that cancer cell. And so what you want to do is you want to starve the cancer cell because it has a limited number of things it can use for energy, while allowing the body to have free reign, because the rest of the healthy cells can use fatty acids and ketone bodies as fuel. So your press pulse method, and I'm going to turn it over to you because you're the expert. Talk about what you're pressing, what you're pulsing, and then let's get into some of the other things that are being proposed, things like hyperbaric oxygen and other things that can accelerate the rate at which you clear the cancer cells.
Yeah. Well, the thing we have to also recognize when we say we have to look at the two types of energy that we know about in cells. One is, as you mentioned, oxidative phosphorylation, which is based on Peter Mitchell's chemoosmotic hypothesis with a proton gradient of hydrogen ions that are dissipated, and it's like an impeller, and they can synthesize large amounts of ATP inside the mitochondria using the TCA cycle together with the inner membrane of the mitochondria. This is a very sophisticated structural generation of energy. That's called oxidative phosphorylation, because oxygen is the acceptor of the electrons, and the waste products are CO2 and water. So that's and every time we exhale, we have CO2 and water, we bring oxygen in, and this is the combustion of energy inside of our bodies. And essentially, we're just breaking down carbon-hydrogen bonds from the foods that we ate. Those carbon-hydrogen bonds contain energy, and that energy comes originally from the sun through photosynthesis. So we're just simply recapturing the energy of the sun by burning the food, and the waste products are CO2 and water, right? But the other form of energy is the energy that we had before mitochondria developed billions of years ago, which is fermentation, energy without oxygen. And that's called substrate level phosphorylation. And that's where a phosphate group is moved from an organic substrate onto ADP. And that can happen in the cytoplasm through the glycolysis, the Embden-Meyerhoff-Parnes pathway, with pyruvate as the end product, which then would go into which would go fully oxidized in the mitochondria under normal conditions. But if the mitochondria are dysfunctional, it's reduced to lactic acid, and that's dumped out. But the energy is coming from substrate level phosphorylation. But what we found in cancer cells is that you can have another substrate level phosphorylation reaction inside the mitochondria, independent of oxidative phosphorylation. So for a lot of folks, they get misinformation because they say, oh, I have a cancer cell. He's taken in, the cancer cell is consuming oxygen and making ATP. And they say, well, see, it looks like Warburg's rung. It's making it. They're using. You're right. They use the energy. They use the oxygen to make ROS. And the ATP is coming from glutamine through the fermentation mechanism. It's unbelievable. So now we know what these cancer cells are doing to stay alive. But you're right. So we have two forms of energy. the substrate level phosphorylation, the primitive form of energy. And then we have the mitochondrial oxidative phosphorylation in the cristae of the mitochondria. So when we look at under the electron microscope, and I did this in the paper that I published on the origin of ATP and cancer, first thing you have to do is say, okay, if you're telling me mitochondria are abnormal, what's the evidence for that? So you go back and you take every major cancer that we know of, breast, colon, all of the different cancers, lungs, and you look under the electron microscope at all these pathologists have looked at for decades, back in the 1950s, 60s, and 70s, when the electron microscope was first being developed. Everybody wanted to look at stuff. They have the beautiful pictures of mitochondria. There were no cristae, very few of them. Well, how are you going to make energy when the very organelle you're looking at is busted and all falling apart? So you look at that and you say, well, that can't. Structure determines function. If the mitochondria is a very complex structure, if you look at it under the electron microscope, and when it's abnormal, the structure is very dysfunctional, a very abnormal structure. So you know automatically that organelle is not going to be able to produce energy as efficiently as a normal cell that would be in that tissue. So already you see this. And then people still don't believe it. They believe, and this is one of the great things in biology. When we talk about cell biology, most cell biologists look, oh yeah, that doesn't work. It's dysfunctional. But when you show the same picture to an oncologist or a cancer guy, oh no, it's completely functional. What's going on here? So we have two worlds. We have the world of oncology that doesn't see changes that everyone else sees. And they're all doing things, and it's just unbelievable. So, but we saw that. And then the other thing that I just published this year, 2024, for the first time, the other second big thing we see is the accumulation of lipid droplets in the cytoplasm of cancer cells. So every major cancer that you see with abnormalities in the number, structure, and function of mitochondria, they're all storing lipid droplets. And the field of oncology says the cancer cells are storing the lipid drops because they need so much energy, they're going to burn the lipids. And the answer is no. They're storing the lipids so they don't make reactive oxygen species from them because they're going to die if they do. So they store the lipids in the cytoplasm, which is a marker now for inefficient oxidative phosphorylation. So those are clear markers to tell us the mitochondria aren't working well because they can't burn fatty acids. And if you try to get them to burn fatty acids, you're right, they explode with reactive oxygen, and that kills the cell. So to protect the cell from death, they store the lipids in these droplets, not because they need them for energy, but because they don't want to die. It's just
so is this the reason why? Is this the reason why people are adding the hyperbaric oxygen to the press pulse method? Is because when you have the cells in a situation where oxygen is actually going to be very destructive to them, you're increasing the pressure of oxygen from, what is it, about 3 pounds per square inch at atmosphere?
Yeah, 2.5.
Okay, and you want to increase it up to,
No, we go up to about 2.5
atmospheres.
Yeah.
Okay, so you're up at around 7.5 to 8 pounds per square inch of oxygen, partial pressure of oxygen. But what you're doing is you're really driving more oxygen into the bloodstream so that those cancer cells, when they take up the oxygen, they're more prone to literally just popping.
Yeah, but you got to realize what's cancer protected against ROS by having a powerful glutathione and a superoxide dismutase antioxidant system.
And melatonin.
Yeah, and melatonin. But we know that that's coming from the pentose phosphate pathway, the glutathione through the NADPH. But the glutathione is made up from glutamine. Glutamate is one of the key amino acids in the glutathione. So and the glucose is driving the pentose pathway to make glutathione, and the glutamine is giving us the glutamate. So protecting us against the ROS. So when you take the cancer patient and put them in a low glucose ketone index where glucose is low and ketones are elevated, the cancer cells now become so much more vulnerable to oxidative stress inside the hyperbaric oxygen chamber. So their antioxidant shield has been perforated, And now they become even more susceptible to oxidative stress because their protective shield is now being pulled down.
So
And
let's just break this down. So what you're saying is that because the cancer cells have a high dependence on glucose, and you can withdraw glucose from the system by simply just getting into ketosis, therapeutic ketosis. That you want to do constantly.
Yeah.
Then, because glutamine is not an essential amino acid, it's produced in the body. There are pharmaceutical agents, I think one of them is called Don, that you can go on briefly. During that period of time, you will suppress glutamine synthesis.
Yeah.
And if you do it at the right time, the glutamine levels drop. You can't make glutathione.
Yeah.
Which is a consequence. You can't, not only can't you use the glutamine for energy production,
right,
but the cancer cells can't lose their shield, as you're saying, because they're not making the glutathione, which is, what is it, glutamine or glutamate, cysteine and glycine. And glycine. So that trio of amino acids that forms glutathione is now dysfunctional, is not working, and now you become susceptible to the oxygen.
Right.
Even more susceptible.
Right.
Okay. So now I understand where the hyperbaric oxygen comes in.
Yeah. So, but that's why we, in the press pulse therapy, we recommend patients being in a low glucose ketone index, which is going to deprive the pentose pathway of carbons to make glutathione. So, and then you're going to start, the shield is going to start coming down. And what Don does, Don blocks glutaminase, which is the metabolism of glutamine to glutamate. So you block that step. So you're shutting down the availability of the glutamate. The glutamate's not going to be there either. And your energy is not going to be there. So these tumor cells, man, they are just absolutely sitting ducks for this process. And the rest of the body is burning a ketone body, which is considered a superfuel, which actually reduces oxidative stress in normal cells, making them energy more efficient. So your body is now becoming a super your normal cells are becoming super healthy, while the tumor cells are becoming super weak. And your body now is taken up with little glucose. And the organs that take the glucose is the brain and the muscle. So you put a little exercise in. Muscle does not share glucose. It's an evolutionarily conserved process. You've got to run away from the lion when he's chasing you. So you're not going to be sharing your sugar with any other organ because it's an evolutionary conservation system. And what little circulating glucose might still be present, the brain's going to suck it up. What happens to the tumor that needs the glucose? You're taking away its glutamine, you're taking away its glucose, you're taking away its protective shield. It's going to die. And we know it. We're tearing apart. The angiogenesis goes away. The inflammation goes away. The body starts getting healthy. The immune system comes in, clears up the corpses, makes a scar or whatever it has to do, cleans up the microenvironment. The acidification that used to prevent the immune system from attacking the tumor, the acidification was coming as waste products from glucose and glutamine. Lactic acid and succinic acid. They're not there anymore. The immune system now, carte blanche, to come in and get rid of the tumor cells. This is unbelievable.
Is there then a value therapeutically to exogenous ketones potentially in this?
Maybe so. We're not yet sure. But I think, my good friend Dominic D'Agostino puts a lot of emphasis into that. And I think we need to, when we start doing the press pulse therapeutic strategy as a clinical trial on people, we will have groups that will have exogenous and others that don't, but we're going to be measuring internally all the time. So we'll have a more direct answer to that question as we move forward. Once metabolic therapy starts to become incorporated in a serious way into the main, and I don't know how long that's going to take. I mean, these poor guys are still chasing genes and things, so I'm not really sure what's going on.
So you're focusing on, if I understand it closely, you're focusing on glioblastoma as your first target. And I understand it's a terrible disease. It's very difficult to treat. And frankly, the modern traditional way of treating glioblastoma is practically accelerating the disease state. So it's ripe for going after it. But my question is, ketone bodies are generally created in the liver. How will this process work, or how would this therapy work in liver cancer?
Yeah, so that's what when we did our analysis across all different kinds of cancers, every one of them is a fermenter. All of them, every cell in the tumor is getting energy through fermentation. So they can't burn ketones. The ketone is a non-fermentable fuel. So we can marginalize almost every cancer because they're all basically have the same common metabolic problem. They're dependent.
But if you're relying on the liver to make the ketones.
Okay. Yes. No, they would burn fatty acids.
Okay.
Liver burns fatty acids, and they store the liver hepatomas loaded with lipid drops. So they're not. they're suffering. You're right. They make ketones. They can't burn ketones, but they burn fatty acids, and they can't use that.
And how do you stop the gluconeogenesis from the liver? I mean, the liver is a source of a lot of sugar because they can actually make it. They can reverse the process, of glycogen synthase. Do you have to exercise? Do you have to burn off that?
Yeah, they can never replace the amount of sugar you would get from your diet. They can just up because you need some glucose to run the erythrocytes in the bloodstream. Because they don't have any mitochondria nucleus. They work purely on glycolysis. So, and there's billions and billions of these erythrocytes. So, and they're going to be the gas exchange carriers. So you have to keep those cells, and they can burn the smallest amount of glucose fermentation wise. So, so gluconeogenesis is making glucose predominantly for the muscle in the brain and a little bit for the erythrocytes. So the tumor's the last one on the line to get a little suck of that. So that's why exercise becomes really important. Yeah. Because if I'm exercising, I'm taking glucose out of the bloodstream. The brain is taking it. The erythrocytes. Where's this going to come for the angry tumor? He ain't going to get it. He ain't getting it.
So this really points to just how important muscle quality and good quality skeletal muscle to burn the glucose is on an ongoing basis.
Yes. Absolutely. Oxidative respiration, muscle burning. You put yourself into a low ketotic state, and then you come in with the drugs that go after the glutamine, and you pulse them. And Don is the most powerful we have ever seen. I mean, there are others out there, but nothing like Don. Don is a dirty drug. It hits multiple because there's families of glutaminases, and the industry likes to patent stuff to make billions of dollars. And I've tried some of these things, but if you block one, it's like whack-a-mole. You block that one, but the other one gets upregulated. Don is beautiful because it shuts them all down simultaneously. And they took Don off they don't use it. They said it was too toxic, and it was toxic, but not like Red Devil and cisplatin and some of these crazy things they're using now. These are poisons, man. They're brutal. But the other thing about Dawn is they never used it in the correct way. You're not supposed to use it as the primary source of treating cancer. It's supposed to be an add-on after you choke hold the glucose. So you got to know how to use the tools that you have in the tool chest.
What's the amount of time stay on? What's the amount of time that you have to stay on the Dawn in the pulses? I mean, how frequently do you do it? It's like once a week. Is it for a couple hours?
That's cutting edge right now. That's what we're working on.
Okay, you're working on
it's called non-sexy science. It's called dosage, timing, and scheduling. Yep. And all it's good for is keeping people alive healthier for a much longer period of time. But it's called non-sexy science. You can't get funding for that stuff. They want to know what the signaling cascade is and which gene is turned on and turned off. Hey, listen, that's great and wonderful, but we're trying to keep people alive here. You think the guy with the brain tumor or the liver cancer or the lung cancer cares about what signaling pathway? He wants to know what's going to happen. How long can I live in a healthy quality of life? That's what our interest is.
And he doesn't care if the pharmaceutical industry is making $1 billion off it. He cares about whether he's going to be around.
That's their personal problem. I don't know. Our job is to keep people alive healthy. And right now, but I have to be honest with you. Yes, doses, timing, and scheduling is what we are working on now. And ultimately, when we achieve that, and that's going to be trial and error in the clinic as well, because every person is a little bit different metabolically from every other person. So again, you have to look at each person as an entity, their age, their sex, their body weight, all these different things. What other comorbidities might they have? And then you have to design your therapeutic strategy around that individual's personal physiology.
Now, one of the things that we talk about a lot on this show is about the autonomic nervous system and how it influences a great number of things that people don't typically think of. And one of them is metabolism. And so I want to first get a question, which is, what's the role of calcium in mitochondria and in the dysfunction? What do you see in cancer cells that have dysfunctional mitochondria with respect to calcium levels within the mitochondria?
Very few people ask me that. It's like an essential issue because the signaling cascades inside the cell are regulated by the inner membrane of the mitochondria, the proton motive gradient. Mitochondria can absorb calcium, and they can they regulate calcium signaling inside the cell.
Big buffer.
And the cell cyclins, that control mitotic division and how fast and slow the cell, are all linked to the calcium gradient inside the mitochondria. So when that gradient dissipates from a variety of insults to the structure and function of the mitochondria, calcium signaling is dysfunctional, unregulated. So the cell now begins to not know my major controller is losing its grip on my existence. So the only thing the cell can do is fall back on a default state, which is proliferation. And don't forget, when we have liver regeneration, this is very interesting, because when you regenerate liver, they grow faster than a tumor cell, but they're not dysregulated. It's a highly specific regulated growth, and the mitochondria are burning fatty acids and ketones, or glucose is there, but it's not the prime fuel for liver regeneration. Fatty acids are the prime fuel for liver regeneration, meaning that the mitochondria are still in control. They are in control of the division of the liver. And in gut replenishment, in the crypts of the gut, that's driven by butyrate, which is a 4-carbon fatty acid. So neither of these regulated growth signaling systems are burning glucose as a prime fuel. Just opposite of the tumor. The tumor can't use fatty acids or ketones, falls back only on the fermentation metabolism, which is either glucose or glutamine together as a synergistic combo. So the calcium signaling is, in fact, regulated by the mitochondria inner membrane proton motive gradient. That links calcium signaling to a lot of regulation of systems within the cell. So again, that organelle is important.
Calcium also links it to the autonomic nervous system. And this is how
I'd have to look more into, but I can look more into that.
Well, the reason is that I bring it up is because one of the most important receptors that's involved in vagus nerve stimulation and in the parasympathetic recovery state and all that is a receptor. It's a nicotinic receptor. It's the alpha-7 nicotinic acetylcholine receptor. Yeah. And you think of that in the context of the inflammatory reflex, immune reflex, because it's on the extracellular membrane, but it's also expressed on the surface of mitochondria. And it's an ionotropic calcium channel. And so when you activate with acetylcholine, the neurotransmitter, obviously, of the parasympathetic, when you activate that receptor, it helps to regulate calcium levels inside the mitochondria. Now, there's a few other pathways that are involved in it, but interestingly, it's very tightly regulated with melatonin. So when you were talking about glutathione before, I mentioned melatonin. Yeah. Melatonin is a really important antioxidant, but it's also not just for sleep. It's actually also part of the regulation of mitochondrial health through when melatonin levels rise, then the expression of this alpha-7 nicotinic acetylcholine receptor rises on the mitochondria, and it becomes receptive to this acetylcholine. There's actually channels in the extracellular membrane of the cell to bring acetylcholine into the cell, into contact and into the vicinity of the mitochondria. So there's a play here where I think the autonomic nervous system is involved. And we talked before we got online, we talked about the fact that some of your work originally was in epilepsy and the possibility of vagus nerve stimulation being involved in the regulation of liver function and in immune function and I think in mitochondrial function. So I think, one of the things that I've looked into in terms of cancer is when people are in a highly sympathetically active state, they have worse outcomes in cancer by orders of magnitude.
Yeah. Wow.
So if you are in a parasympathetically dominant, sort of that rest, digest, restore, recovery state, you have a much better chance of surviving cancer and getting through it. And I think that there's a relationship here between your autonomic nervous system function and this mitochondrial theory of cancer. I think they go hand in glove.
Yeah, I think that's a very exciting and very important connection. We have not yet dug deep into that, what you just mentioned, because we're still proving the point that cancers cannot survive without fermentation. But once we start the clinical application of press pulse, these concepts of what you just mentioned now become add-ons and facilitators of a better, faster, positive outcome for the patients. But we haven't jumped there because we're still dealing with the foundational concept of what cancer is. It's a mitochondrial metabolic disease. Once people realize that, then the path to a much more effective outcome with the sympathetic nervous system and how we can manipulate that and even make the outcome better, it's going to be beautiful. But we're not there yet, because we haven't even started with the exception of a few folks that I've published. We're still in a very infant stage, in a very early stage of this whole process. But I look forward to these kinds of connections that you speak of, and how effective they're going to be when we really know how to push this system in the right direction.
Well, let's talk a little bit about how well this is being deployed already, because I know that you're talking about setting up the real major clinical studies and doing some of that boring science that has to come before you can run the big clinical studies. And I know that the plural of anecdote is not evidence, but there are a lot of people who are following your work and who are even advocating for it. There's the Hippocrates Foundation, I think, or Hippocrates Research Foundation based in Texas. I've heard a lot about what they're doing. I mean, how many patients at this point are you aware of? And I know that you're not a clinician. You're not asking people to go on this therapy. You're just looking to see it tested and studied. But how many people are you aware of that are doing this already?
Yeah, there's a lot of people doing it. I'm starting a little registry because I give out my kit of information to folks, and I just ask if the information helps them, consider making a donation to the Boston College Cancer Fund that I get the money for, or Travis Christofferson's Foundation for Cancer Metabolic Therapies. They're the prime supporters of our work. But I only ask them to do that if the information helps them. I mean, I don't charge anybody any information. For me, success is their outcome. Okay. And if they want to throw a few shekels in our direction, we're always appreciative of that. But I get, so I give these kits away. I don't know. This guy calls me up. Oh, I got advanced metastatic this or that. And I give him the kit. And then about a year later, he emails me. He says, I'm just still alive. I'm doing really well. And, I had that big tumor. And the doctors are always miffed. They don't know what happened, why I did what I did, and how the outcome came out. And so I keep a registry of those folks. And I get a big kick out of it. And I said, geez, I thought this guy was a goner. I didn't know how he could possibly survive, and he's feeling great. So Maggie and Brad Jones are doing a big documentary, The Cancer Revolution. And they're making a documentary movie because Maggie Jones had metastatic lung cancer to the brain. And she went on her metabolic therapy, and she's out 10-year survivor now. Mm-hmm. So Brad is a professional documentary movie maker from Hollywood. And they're making this big movie, and they're collecting a registry of all these folks that are supposedly terminal, right? Terminal this, terminal that, lung, colon, brain. And they're all, I'm just still alive. Yeah, I'm doing well. And, all this kind of stuff. Of course, that's great. But I also have to publish peer-reviewed case reports so they know what they did to survive. Mm-hmm. And I think that's... And I'm telling you, that's tough, man. You try to write a human case report, man. You got all kinds of loose ends hanging around. Why did you eat that at that time? Don't you know what it did to your GKI and all this? But my students and I, and we work together. I'm working with physicians because I need to get the data from them. I need the patients to be compliant, so they collect the data from me, and I can publish these papers and let the cancer field see why this terminal guy is still alive. What did he do? We were very saddened this a couple of weeks ago, or last week, actually, 2 weeks. Our big brain cancer survivor, Pablo Kelly, passed away, a 10-year survivor who used for glioblastoma. He had no radiation, no chemo, no steroids, none of this stuff. He did metabolic therapy by itself. They said he had an inoperable brain tumor. After 3 years, they did the first debulking surgery. He kept his GKI nice. We published it 80 months. He had 3 surgeries over a period of 10 years on this tumor. He was never cured. He got married and had several children. He was just using metabolic therapy to manage the tumor with just ketogenic metabolic therapy. But then he went in. We were having a big conversation, just like we're having right now. He was having a good time. We were talking. He looked healthy. He was great. And the next day, he said, "I'm going in for." There's a small part of the tumor they think they can get out, and they went in there. And he came out of the surgery, his thumbs up, smiling on talk. He had a massive cerebral hemorrhage and died the next day. 12 hours after he completed the surgery. This guy was healthy as he could walk. He had. I mean, can you imagine surviving glioblastoma for 10 years and still being healthy, and then having an inoperable tumor operated on 4 times? And the last time he didn't die from the cancer, he died from a cerebral hemorrhage as the result of a problem with the surgical procedure. So that was because he was our poster child for how long you could live with a glioblastoma using metabolic therapy. But he did not die from the cancer. And I don't know if he didn't have that last surgery. I don't know how much more he would have. Because he was out 120 months, which is 10 years almost to the day that he was diagnosed. So it was kind of hard because he had podcasts. But we still have Maggie. We still have a lot of folks out there with lung cancer that are still doing very well. So I think we're going to see a lot more of these folks surviving. But who's going to write up all the case reports? I'll have a stroke writing up the damn case reports. So
let me ask you a question about these folks. How difficult is it to get yourself into therapeutic ketosis? And I understand that for the average healthy person who's walking around and likes to go to Baskin Robbins and get an ice cream cone, they're not going to want to do this. But for a person who's got cancer and it's inoperable or terminal, chances are they're willing to do some pretty extreme things. Is it that extreme?
No, it's not that extreme. It's kind of a frightening. People get, what am I? Well, you have your choice. You want radiation? We're never going to stop anybody who wants radiation and poison. That's not what we're doing here. I'm not telling you don't do that. What we've learned though is we can go on a 0-carb diet and bring down the GKI. So you can eat fish and meat, and if you're a vegan or whatever, you try to get your GKI down, but eat 0 carbs. And after about 14 days or maybe even 3 weeks at the most, you would see your GKI dropping significantly. And then you can easily jump to water-only fasting for a few days. And that's when you come in with the glutamine targeting drugs. So it's not so, but if you try to go, it's not so traumatic, but if you try to go cold turkey, it's tough because the brain is addicted to glucose. And it's like trying to withdraw from nicotine, alcohol, or whatever, opioids or whatever. It's a very difficult, your body reacts in a very strong way to this. But if you go 0 carb, and even that can, after about 7 days of not having a bagel or something sweet, hey, you're looking around for something sweet. But you have the glucose ketone index that's telling you you're on the right path. You're on the right path. But I've had people tell me, I've been eating no carbs and I just can't get my GKI down. I said, well, stop eating, just drink the water. And invariably it drops.
I was going to ask about fasting because I think obviously the effect of fasting is to eliminate the excess carbohydrates, to have a very positive effect ketogenically. So fasting is a tool that you're using in the press pulse method, yes?
Yes. Well, you can do it with 0 carb diet. The goal is you use the GKI to determine the zone. So, and that can happen if you're, again, a vegan, a pescatarian, a Mediterranean, whatever it is, we don't care. Whatever you want to eat. And if it says, well, I can't get that, well, stop eating that. Try eating something else. It's not that difficult. But you use the number that from your body's number is telling you what you need to do. And then that could be different from you or somebody else. You got religious differences, you got cultural differences, you got all these differences that people have. And then but bottom line is you're still a machine. You're still a bomb collimator burning stuff. And you can get down into those zones.
How do you get a list of foods that literally have 0 carbs in them? Because you go to the store and you think, oh, I'm going to get some beef jerky. Okay, just beef. It's dried beef. But you look in it and they got a flavoring in it, and there's 6 grams of sugar in it.
Yeah. Well,
how do you?
Well, you have to be conscious of that. And what we did in the epilepsy world, we had big lists of low glycemic foods. Like we realized that grapefruits could provide you with massive amount of vitamin C and doesn't spike glucose. So we learned that there were foods, avocados, and there are foods, and there's a glycemic index associated with each type of food, and a patient would choose whatever they like from the list of low glycemic foods, and they would monitor their glucose ketone index. They're the ones they're monitoring their own. They know what eventually, and then once they know their GKI, sometimes they know they can feel themselves coming out of the GKI. And they know when they're in the without even have to measure it because they've done it so many times. So you're turning your body back into the Paleolithic lifestyle that we had. As I told many, many folks, we are still Paleolithic man living in a modern technological society. And the cancer, diabetes, obesity, all of these are the result of a rapid change in our environment. So we are still evolved over 1.5 million years in a hunter-gatherer, thousands of years in a very glucose minimal environment. And then within the last, 50 or 75, 100 years, we're all of a sudden exploding with all these different diseases as the result of our technology giving us the kinds of foods we always wanted because they were so tasty. Now they're on every corner, Dunkin' Donuts and all this stuff. And they have obesity. As I said, obesity is evolution in action. Those guys that are obese today are the descendants of the survivors of famine.
Right. That's exactly right.
Right.
What role does the microbiome play in this? You mentioned butyrate earlier as a short chain fatty acid that's going to help support liver function. And we know that microbiome is heavily involved when it comes to leaky gut syndrome, activation of lipopolysaccharide.
It's kind of ambiguous to me. I mean, as long as you have, I always find that in cancer, that we talk about the microbiome. Nothing could be more destructive to the microbiome than radiation and chemo. So why are you talking about wondering, keep your microbiome? You're poisoning and radiating somebody. The poor guy's got diarrhea. He's got all kinds of problems. Well, in that case, we can discard the microbiome. See, with metabolic therapy, you will use the kinds of things that you need to manage the cancer and keep your microbiome in the right spot. So it works for everything. But it's a hard sell right now. It's a hard sell.
Absolutely respect your time. We want to thank you so much for joining us today. I just want to quickly recap. Mitochondria is key, right? The mitochondropathy is the initial pathway to this. It's glucose. That's the initiating factor. Excessive glucose in the bloodstream. That's the initiating factor. And we have to be careful because it was called the oncogenic paradox by Albert Szent-Györgyi.
It was like how many different things provocative agents can cause cancer through a common pathophysiological mechanism. And what I did is I defined the common pathophysiological mechanism that was always elusive to others. Viral infections damage mitochondria, leading to compensatory fermentation. Intermittent hypoxia, carcinogens in the environment, systemic inflammation. All of these things can impact people in combinatorial ways, affecting mitochondrial function in a particular cell of a particular tissue. So, but the outcome, of course, is a cell driven by a fermentation metabolism. Right. So its origin can be different from one person to the next, but the outcome is always similar.
And it's that fermentation of either glucose.
There's a replacement of oxidative phosphorylation with substrate level phosphorylation using the fuels of glucose and glutamine. And some tumors are more glucose dependent than glutamine, and some are just the opposite. But in press pulse metabolic therapy, we take them both away. We know they can't survive. And we tested every other metabolite. We looked at all the amino acids. And that's the way we do this. We starve the cells. We take everything away from them, and then time how long it takes them to die, because there's nothing there but salt solution. When there's no food, they die. And then we add food back and find out what perks them up. And then you immediately know what's keeping them alive, and then you can take that away and kill them. It's really amazing.
Pretty easy observations when you have the right mindset as to what you're looking.
Oh yeah, we've blown this cancer thing up to make it a hopelessly complicated disorder. It's not so hopelessly complicated. It's actually relatively simple that we as humans have made into a monstrosity. But the approach to management is not so easy. It might be easier to sit there and get infused with a poison, or sit there and get irradiated. You're not doing anything. But when you are battling your cancer with metabolic therapy, you are in fact battling your cancer. Mm-hmm. The success falls on your shoulders. So people need to know that they get empowered to know that their outcome is dependent on their level of motivation to stay with the metabolic therapy. So this is a completely different strategy for managing, but it will work, and it will drop the death rate by 50% in 10 to 15 years.
I found it very interesting when I first really learned about your theory. One of the things that struck me was the fact that 100 and some odd years ago in Philadelphia, there was a physician. I think he was maybe even been the leading physician in the hospital. He stopped everybody doing any work, brought them all into the surgical suite. He wanted them all to witness a case of lung cancer, and he told them all it was very likely that they would never see one in their careers. Lung cancer. And think about the amount of smoking that was done back then.
I know.
But there was no sugar.
Yeah.
And the amount of sugar, Dr. Gundry talks about the fact that a 16-ounce bottle of Coke has more sugar in it than the average person in 1850 had in a year.
Yeah. Yeah.
So 55 grams, by the way, 55 grams in one 16-ounce bottle of Coke.
Oh yeah. Oh no, yep. we never had, but sugar is not a carcinogen. Sugar creates systemic inflammation, and especially high fructose. Yeah, and that contributes to the problem. And once the cancer starts, then the sugar is a prime fuel.
Yep.
So, but any event, we're just remember, we as our research depends a lot on philanthropy and donations. We actually acknowledge in our papers people who want to recognize to be a part of the change. So we put, if you see some of my papers, you'll say, who are all these guys? They're the ones who are contributing to our research.
They're making it happen. Yeah,
they're the ones making it happen. And there's a lot of folks that just want to jump and feel that they're going to do something really big for people. So that's what keeps us going.
I can't imagine there's too many other projects that are as effective and as promising as what you're doing. So I would encourage anybody listening to this, not only to, as Dr. Habib often says, if you get to this point in the podcast, and we'd love you to share it with somebody that you think needs to hear it, at least one person. But not only that, but in this case, help fund the research. Fund the research, because this is really going to change things. It's not a lot of projects in the world that have the chance of saving millions of lives, and this is one of them. So hats off to you for doing that.
Yeah, well, thank you very much. But I get a little upset because there's some folks out there that are writing books with my name on it that I never wrote, and others are kind of siphoning off, saying I'm supporting these foundations, and I don't. So it's only Boston College and Travis Christofferson's foundation, nothing else. So I want to make people this charity thing and all these. It's upsetting a lot of my students. Because it's not, we need those funds to support our research, not pad somebody's pocket.
Yeah, but, everybody be very aware if you're
it's like money. Anything, all this kind of stuff. Any event, listen, I'll let you gentlemen go. Thank you very much for the interview, and I hope your audience learns something about how to manage cancer without toxicity.
Absolutely. Thank you so much, Dr. Seyfried. This was absolutely a pleasure. And GP, why don't you close it out?
Yeah, no, I think this is a great spot to end it.
Yeah.
Thanks everybody for listening, and we'll be back next week.